So thank you, everyone. Good morning. Oh, I guess good afternoon or evening, wherever you tune in from. Um, thanks for the introduction, Kelly. I'm Aik, and I'm a R&D scientist at Mission Bio. And today I'm very excited to tell you about the product I've been working on, and primarily focusing on the wet lab part on how you can perform this DNA and protein protocol on the Tapestri platform. So first, I want to just give an overview of what I'm talking about today. I'll give a technology overview of how Tapestri works and how we can incorporate both DNA and protein workflow on the Tapestri platform. And also, I'm going to go through some very exciting data on what kind of analysis you can do with these multi-omic analysis and how it would improve your cell profiling from your heterogeneity sample using this type of data. And at the end, just kind of a couple of slides of basically a teaser of what's coming next. I want to start by showing this slide. Basically, for most people, at least for me before I joined Mission Bio, when I hear Tapestri, I just think of the Tapestri instrument. But I want to point out that Tapestri is actually a complete suite of products. And it guides you basically from the experimental planning stage, doing your panel design, all through your library prep on the Tapestri instrument, and also your secondary and tertiary analysis using the Tapestri pipeline and Tapestri insights. So for today's presentation purpose, I'll be focusing on this box area, basically the sample prep and the library prep using the Tapestri instrument to do your protein and DNA multi-omic analysis. So for those who are familiar with our technology, this will be very simple to you. But for the new audience, I would like to point out that our microfluidic device is using this very unique two-step workflow on the droplet generation. So basically, you have the first step, you do the encapsulation of your cells into your first droplet. You take that emulsion off the Tapestri instrument into a thermocycler, and you do your basically a lysis heat treatment, and then followed by a heat inactivation of some of the enzymes. And then you take that emulsion and put it back to your Tapestri instrument for a second drop formation. And this second drop is going to pair you up with your cells to your barcoding beads that carry the cell identifier barcodes, and also your PCR reagent mix. And once this second drop is formed, you take the emulsions off the Tapestri platform again, and you do your PCR amplification. So the concept is very simple. For the protein, so this is basically the general basic workflow for the DNA sequencing. For protein, there's very, very minimal difference in terms of workflow on the Tapestri platform itself. So the major differences is we have before even using the Tapestri, we have a sample prep guideline, and I'll go through that on the next slide. And then after that, you load your cells like you would into the Tapestri. And then one minor change is during the cell barcoding step, once you add your reagent mix, you have one extra primer that is specific for your protein libraries. So that's the only change on the Tapestri platform. And then for the PCR amplification, you have basically very similar cycling condition with slightly different number of cycles when you do protein and DNA. And then afterwards in the library prep stage, so basically post Tapestri platform, then there's a separate library prep guide. And I'll go through that in these slides. So to do DNA and protein, it's an antibody-based approach. So if you have done any FACS or flow cytometry staining, then you're very familiar with the cell staining protocol that you can just pick up this cell preparation protocol very easily. The antibodies are conjugated to oligos. So this oligo contains tag specific for each antibody that will tell you where this DNA sequence is from in terms of which antibodies belongs to. So basically, we treat the cells with this antibody panel, and then you wash the unbound antibodies away, and your remaining cells suspension is what you would load onto the Tapestri platform. The cell loading number and concentration is the same as before. So no change on the number of cells loading into the instrument.
Aik, we have a question from the audience. Is the protocol the same as CiteSeq? Basically, what are the similarities and differences between the two techniques?
In terms of the cell state, oh, okay. In terms of technique, so well, there are some differences. Basically, the main part, we're getting genotype information out of this, right? So CiteSeq, you're doing protein and RNA, so you're not getting your direct genotype information. And for this protein and DNA, you get your targeted DNA information where you can do SNP analysis and CMV analysis. On the protein side, in the grand scheme of things, pretty similar in terms of what kind of protein data you'll get out from this workflow.
Great, thank you.
And the cell staining protocol, we do have a specific cell staining protocol that we're currently actually updating, but the premise of cell staining is very similar regardless of which platform, which technology you're using. Basically, you incubate the cells with some kind of cell blocker, and then you add your antibodies and you wash away the unbound. So those are the basic steps in the cell staining protocol, but we do have a guideline on that. And if you're running this protocol, you should use our guidelines on cell staining. So next, so this is basically 3 to 6 on this slide is basically the diagram from the previous slide. So this is basically where you'll interact with the Tapestri platform. So from the stained cells, your cell suspension, you load it into the Tapestri instrument, you do your first droplet encapsulation of the cells. Here now, this figure is more detailed. You have your cells, you have your protein bound to your antibody bound to your cell surface protein. These are loaded, encapsulated in your first droplet. Then you take this drop emulsions out, do your digest and lysis of the cells. Now you free out your DNA and your antibody tag into this droplet. Now these droplets get encapsulated into your second droplet here. And your second droplet, you have the barcode beads and also your PCR reagents. And this is, like I said in previous slide, this is where you add your one extra primer that's specific for your antibody library prep. Now here, then you take this emulsions out and do your amplification. Basically, it tags your targeted DNA with the cell's barcode, and also with your antibody oligo is tagged with the cell barcode. So now you know where all these your targeted DNA and your antibody tags are, which cells they are from. And next, so this is slight alterations on the protocol post Tapestri platform into your library prep. So in this stage, step 7 is actually very important. And in the protocol, we actually put like red warning label. Just remember, it's because now we're separating the protein and DNA product by size. So after barcoding PCR step, usually there is an impure beads purification of your DNA library product. in the normal protocol, or in the DNA only protocol, you have your DNA product bound onto the impure beads, and then you take away and throw away the supernatant from the first wash. But in this step, your protein product is actually in the supernatant. It's a smaller size oligos compared to your DNA product. So you actually save that, and that goes into its own purification step that is biotin and strep beads based. For your DNA product, you continue as you normally would, doing your washing the beads with ethanol and then eluting in water. So now after this stage, you have 2 tubes of your barcoding products that will go into library PCR. So this library PCR basically adding indexing. So add your indices of your choice to your DNA or protein library. I'm only showing one here because they're actually very similar. So you have two tubes, but you do very similar PCR steps on it. For the protein library PCR, we recommended higher number of cycles, but the temperature is the same for both. Again, for the DNA product, you continue your library PCR basically the same as the DNA only protocol. So just give some clarity on the antibody oligo that I've been mentioning. So antibodies are conjugated to an oligo, and that oligo contains three main parts. So basically in this gray square here is the capture sequence. So that part is used after you separate the supernatant from your first impure beads purification. That part, it's the complementary sequence of a biotin oligo that will be provided in the eventual kit when we launch. So you treat your sequence with the biotin oligo, and that you add that treatment into magnetic strep beads. So that's how, and then after that, your library will be bound to the beads, and then you can wash the beads, wash away all your PCR, leftover PCR reagents and primers from that. This red sequence here, the antibody tag, is basically a unique kind of barcode, if you may call it. Just basically gives each antibody a unique sequence. So from your sequencing data, if you map to this sequence and you know what antibodies that oligo is from. And then the final part of the structure is basically a priming site for this extra antibody tag primer that you would put into the barcoding step. So that's the only change you make during the Tapestri Platform workflow, is to add this antibody tag primer. I should also add both the capture sequence and the priming site here, the PCR handle. They're universal across all antibodies. So you only need one biotin oligo for your capture during library prep, and you only need one antibody tag primer. And the reason for this is, the reason to have this primer is so that your tag, your antibody tag will be extended to include this common sequence. And this common sequence is what will prime to your cell barcodes sequence that is attached to the barcoding beads. So now during the barcoding PCR step, you have this extension for the common sequence. And then after the extension, you have further extension to make this full length sequence that goes from cell barcode, and then you have your antibody tag sequence, and then your capture sequence for your biotin strep purification. Now, just to show you what you would expect for your library traces, for the protein libraries, all these tags have the same length. So you're expecting a very narrow peak at around 250 base pairs. So this is what you want to see after you do your library PCR and then further impure purification at the end. And then for the DNA library, it depends on your panel. You see more of a broader peak because the DNA, your targeted amplicons, not always have the same size. But you're expecting something around 450 with somewhat a broader peak compared to the protein library. And again, the DNA library should be the same as your normal DNA-only protocol. So that's pretty much it on the review of the technology. I know I went through that pretty fast, but I just kind of want to get to the data side. So Kelly, is there any questions from the audience on the protocol before I move on to data?
No, I think we're good and excited to see the data.
Yeah, it is a very simple workflow, so I don't expect people are confused about it. So the data. One of the most frequently asked questions, or at least to me anyway, people ask me, so how does this compare to flow cytometry? That's the gold standard that people are doing protein analysis. Basically, they have an idea of what protein data would look like on flow cytometry. So this is done in collaboration with Sargil's group at UPenn. Specifically, I want to give a shout out to Olga Shestova and Catherine Cummings, who helped coordinate with me and run this experiment on their site. So they have this multi-parameter flow cytometry data from 5 AML cell lines that they have done before based on these 7 protein markers. And so when we're discussing experiments to run, they ask, okay, we have this data and we want to compare the data. So let's make this, this panel and test it on Tapestri. So this is what I did. I conjugate these antibodies to 7 different oligos, and Olga actually run this experiment at UPenn. So they run this using the MLV2 panel on the DNA side. So that one has 127 amplicons. And here, the color shading of the boxes doesn't really have any significance. It's just easier to draw my attention to when I point it out. So if you go column by column, for example, focus on the blue column, you're tracing the same antibody across different cell lines. And then if you go horizontal, in this case, like for example, the yellow box, you're tracing the same cell line across the different antibodies. So just to trace the, in terms of MOM14, if you look at number 1, which is the Tapestri experiment, it follows this pattern across these 7 analytes, protein analytes. And you see the exact same trend when you follow their multi-parameter flow cytometry experiment. Kind of similarly with the CD117 across all cell lines, you kind of see basically fairly similar trend across each other. For example, the top one here for the HEL92 cells, you kind of see more of a spread of CD117. We kind of can see more of a spread in the Tapestri data as well. Actually, to guide even easier to follow, we put down like a middle line across the plots and kind of label it high, medium, or low compared to both of these experiments. And to be honest, the concordance is pretty high. Actually higher than what I expect. I thought I might see a lot more of like uncorrelated data, but it was pretty good. Most of them are fairly correlated. A few of them, the ones labeled red here, are we get slightly different data compared between Tapestri and flow cytometry. For example, CD110 here, in the Tapestri experiment for this cell line, we found to be low expression. It's kind of like a median expression from their flow experiment. And then I want to point out this one here, the Kasumi cells for CD33, we kind of get like a medium expression level here on the Tapestri, but there's a low expression in the flow cytometry. There's a couple caveats with this comparison, which will actually explain why we're seeing a few differences here and there. First of all, these experiments are performed like months apart, right? So the cells are also not synchronized. They're probably in a different passage number. So we don't expect complete full concordance between each other. Another main point is also I have to make this panel with whatever antibodies I had at hand at that point. So they're actually not the same clones. That's why I said earlier that I didn't expect such a high correlation between both of these experiments. But overall, this is very promising. What you can produce on flow cytometry, and you can produce on Tapestri as well. And I might also add that this is all these experiments, all 5 cell lines of the protein staining and then sequencing of the tag are done along with the DNA sequencing in the single cells as well, all within one experiment. So there's a lot of parameters here. It's just all fits in one experiment.
Yep. Sorry to interrupt you. We had a couple questions about the protocol.
Yeah.
So what is the optimal cell number to load for these experiments?
Would it be to before staining or after staining?
Both would be great.
Oh, okay. So so it depends on your cell line. Some cells are, some clinical samples we found that it doesn't like to be out staining and washing. So we tend to start with slightly higher on those in case the cells kind of die during staining. But generally, I'll recommend between 500,000 to 1 million. That's the range for staining input. Now, after your washes and staining and wash to load on the Tapestri, it's the same recommendation before. So you load around 100,000 cells in the concentration of around 3,000 per microliter.
Great, thank you. And then one more question about hashing. Is that possible?
Yeah, so you're ahead of me. That's actually one of my last slides. So I'll get to that.
Great.
Cool. Okay, I think I'm going slower than I wanted, so I'll try to speed up. This slide just to show that with multi-omics analysis, you can actually get better resolution of distinguishing your cell lines in your experiments or your sample type in your experiment. In this case, we have 4 cell lines. We use 6 protein targets, 127 DNA amplicons. If you look at just this SNV data, I also want to point out that this is one of my earliest experiments that I've done. I don't sequence the entire experiment, and I also sequence quite shallow on the MySeq. So I don't expect super great resolution from each of the omic. So with that said, when you look at SNV, from the 4 cell lines, you get 3 clusters. You see like K562 and Tomoy kind of clustered together. When we look into CNV, some kind of actually slightly better separations, because even though these 2 cells cluster together, but you can see the separation a little bit from each other. And the protein t-SNE looks like this. Is you kind of have some clustering based on the cell lines. And this is just off 6 protein targets. But when you start combining the omics together, so here you're doing CNV and protein together, you're getting a really good separation already. You see 4 distinct clusters. And when you add in all the information together, CNV, SNV, and protein, you get really good clustering out of these 4 cell lines. So even with shallow sequencing, and again, the experiment I'd done wasn't planned to actually get good biology out of it. This is just cell lines. I was just testing my protocol. So even without proper selection of panel that should, distinguish your sample type and for the protein and DNA panel sites, just because we're getting so much information out of one single experiment. And when you combine all these omics analysis together, you get very, very good resolution of your cell profiling. So it's just another example. In this one, it's a better sequence. It's a sequence deeper. So in this case, I have 3 cell lines, 16 protein targets, and 127 DNA amplicons. A protein alone, you can already separate out the distinguished major cell population. And then by DNA data here, the hierarchical clustering, because these are cell lines and we already know the truths of their SNPs, we can, based on their SNPs clustering, we can tell which is what cell lines, which is labeled here. And I projected the SNV called cell type back to the protein t-SNE, and you actually see it basically mapped directly up to each main cluster as the cell type. So so here is great because you know that your SNPs can tell your cell types, and it matches up with the main group software protein t-SNE. And but actually you can already call your cell type from the protein expression because we know what cell surface markers are on this cell type. So for example, Jurkat cell, which is T cell line, you expect them to be CD3 positive. So it matches up with what the SNV assignment for the cell lines are. Same with the Tom one, which is more B cell origin, you get CD19 and 24 positive. And then for the KG1 cells, we know these cell lines are higher expression of CD44 and CD34 compared to others. So we're seeing that here. Another way you can represent this data, it's kind of with this plot that's more similar to a FACS plot or a flow cytometry plot, which is the pairwise scatter plots. Because everyone look at these plots, they know how to interpret it. So with our Tapestri protein data, you can generate these plots as well. I'm just going to point out, for example, this, I plotted CD90 against CD44. And with these 2 markers, we can already distinguish 3 cell population. For example, here you have the CD90 high population. So you go up to CD90 high, which is this population here. And you know that's calling out your Jurkat cells. So it's here. And then you have your CD44 high, which is this population here. That is your KG1 cells, which is here. And then your double negative population here, which is your CD90 negative and CD44 negative, which is this cell line, with this cluster here. And that will be your Tom1 cells right there. So there's a lot of plots you can generate with this multi-omic data. We're still learning ourselves. We're looking at new data coming in every day to try to figure out what's the better way to represent this multi-omic data. So I've been focusing on cell lines. I want to show at least one example on true clinical samples. And this is done in collaboration at one of our beta tester site, Chris Horrigan lab at the NHLBI. And we use 6 protein markers here. So it's a small protein panel. It's only 6 of them. They run this in the AML V2 panel for the DNA side. And this patient sample is actually very homogeneous. So if you look at the SNV, you have this huge cancer population and just a small population of normal cells. Similar with CNV, you don't get much resolution out of it, but just with that 6 protein markers, you actually get, again, you get a big cancer populations and you get 3 smaller populations here. So at first we were wondering what these are, but when you look further into the protein expression, first I actually want to point out I truncate this part. So this made the main cancer population is 4,700 cells. So if I include the entire thing of the heat map, it's really hard to see this part of the smaller cell population. But this is just a small representation of the main population of the cancer. And so these population here, we know that from the SNV and CNV signatures, they are part of the cancer clones. So we're calling this a cancer subpopulation because they have the same clones. But if you look at the protein expression marker, it separates out from the main group because it has elevated CD11b expression and some elevated CD38 expression and decreased CD90 expression. So this is a small 180 cells out of over 5,000 cells that has this signature, but it follows the clones of the main cancer population. And then for your healthy cells, even within that, we're able to separate out two small population. So the bigger of the two population we know is T cells because they're CD3 positive. And then a very small population is only 30 cells. So this is less than 1% of the cells are CD19 positive. So we know that's B cells. So you get this resolution of what your cell types were within your clinical samples that you are not able to tell by just looking at the CNV or the SNV alone. So before I head out to the next section, I just want to point out we already have 2 preprints in the preprint server bioarchive. And so I encourage everyone to search for these and have a read. And also, both groups have presented in our Mission Bio webinar before. So I encourage you to go to our YouTube channel, Google Ross Levine and Adam, not Google, I just type in the YouTube search bar, Ross Levine and Adam Bate. You should find our webinar videos that they go deeper into these 2 preprints. So what's next? So far, I think I've shown internally 16 protein markers, and I think in Adam Bate group, Ben DeMarie and Cyril Deli did 23. So another big questions I usually get is how high can you go? We are currently still adding on to that panel size, but I can show you some preliminary data. So this is done in collaboration with BioLegend. I want to give a shout out to David Green, who actually performed this experiment based on my protocol. So in this experiment, there's they spike in some Raji cells into 1 donor of PBMCs. And this is done with 44 protein targets. So we get into the territory that is really, really difficult to run using flow cytometry, or almost impossible, basically. And so with that, so you can do a UMAP plot here showing the different clusters of your PBMCs. And then from the genotype information, we can project the genotype called whether they are Raji or PBMCs onto this protein t-SNE, UMAP. So we know that this population is Raji cells. So here is showing you how the cell types are called based on SNV. So you have PBMCs following this, this different signature of SNPs, and Raji cells have this signature of the SNV. And from this 44 protein marker, we separate out kind of about 8 different cell types that we can see with different signatures based on the proteins. And I'll go into further detail in this slide. Yes. So within the PBMCs, just remind you all these PBMCs, if you just look at the DNA data, it's all 1. They're all from the same individual. It has 1 clone. And then, but in this 44 antibody panel, we're able to separate all these different cell types. You can find out the T cells, the cursor or the crosshair point to the group of cells. And then below this red line, it tells you the expression positive or negative for that specific marker. So we're able to separate out T cells, of course, which is a CD3 positive, NK cells, your monocyte, basophils, eosinophils. So these are getting to very tiny populations. Your B cells, which is CD19 positive, of course, and your Raji spike in, which is also a B cells, CD19 positive, but it has slightly different signature because this is Raji cells is isolated from a patient with Burkitt's lymphoma. So it's tend to have some tumor signature as well. So in this case, it's CD38 positive and CD71 positive as well as for the Raji cell. So within protein expression as alone, you can also separate that from the B cells that is from the PBMC donor. And then another question I guess asked a lot is sample multiplexing or cell hashing, if that's a more familiar term for you. Now, of course, I got that question just today, just within this seminar. So yes, it's doable. It's something that we're still in development. We don't have a timeline or where that we're launching that part of the product, but it's something doable on this platform using this technology. So I did a preliminary experiments using 3 cell lines. I basically multiplex them, this mix of 3 cell lines into 4 different groups. So in essence, I should be able to see 12 different clusters within one single run. So this is just a heat map showing all different data you get out from this run. You get just SNV in, so the 3 cell lines I use, JurCat, K562, and KCL22, each have a distinct SNV signatures, and then it has some distinct CNV signature as well. These parts are my sample multiplexing antibodies. So I can, even within the same cell types, based on the hashing oligos, we can tell them apart, which sample number it came from. So each of these cell types have 4 different groups in it. And now, of course, within the cell, it has your protein panel, your staining from your surface markers as well. So you can get different protein markers from your cell lines as well. So these are just UMaps showing how you can cluster basically on SNV. These 3 cell lines have very distinct SNVs based on this DNA panel. CNV less so, but you can still kind of see preferential space of each of the cluster. It just doesn't separate as well. And then when clustering on protein alone, so using the surface protein and also with the sample multiplexing, so there's 4 different multiplexing oligo, and then using the surface protein to distinguish the cell type. Based on that, we should get 12 different clusters, which is shown here. So each of the color is determine which cell type based on the protein expression, but because each of them also have separate hashing oligos or hashing antibodies, and you can able to separate out this based on your sample number. So yeah, sample multiplexing is possible. There's something, like I said, we're still in development. It's not something we're launching right away, but it is a technology that can be implemented on the Tapestri platform.
Wow, Aik, that's really impressive. Just a quick question about the cell hashing experiment. How many cells is that?
So these are about several thousand, I can't remember on top of my head, but it's a normal full run. So I think I get close to 5,000 cells from this experiment.
Great.
Okay, so that was my last slide. Sorry for go over time, but there's a lot of people helping me and our team in this project. So I want to thank a lot of people in Mission Bio, especially discovery and collaboration team, bioinformatics, development team, and manufacturing. Mentioned before, Cyril and Ben, they gave a lot of feedback into our early development of the product. Super grateful for these beta testers and early access that gives really good feedback. So from these, especially from these 3 groups, they're able to take my very early protocol and basically run into their lab and run the experiments and produce pretty good data for us to just to get real sample. I only show one slide on the clinical samples, but just to get several, clinical sample data, it helps us a lot to understand the product and make changes to the protocol, and also to come up with new ways to analyze the data as well. And then again, we're in partnership with BioLegend, and Kit run a pretty really amazing group. David Green's been running the Tapestri experiment, and Jason been helping with the analysis as well. So I'm very grateful of their help and collaborations. All right, I want to thank everyone for tuning in today, and I'll take any further questions.